Good morning. Today we are going to study about genetics, and we will be discussing the various aspects of genetics, that is heredity, chromosome, chromosomal number, autosome, sex chromosome, sex determination, and many other things to follow after that. Now, what is genetics? Genetics is the transmission of characters from the parent to the offspring. Okay, like if a father has a tall nose, the son also will have a tall nose. Will he have or will he not have? The study of the entire thing is known as genetics. Now, what is heredity? Heredity is the transmission of characters. That is how one organism's character is transmitted by the next organism. In the next organism, that is basically called heredity. Chromosomes are the basic unit of genetics, which basically carries the information from the parent to the offspring. Okay, then every organism have got a definite number of chromosomes. Therefore, human beings, you can see here, has 46 chromosomes. Similarly, you can see onion has 16, dog has 78, and this chromosomal number determines an organism what it should be. Then we will look on to autosome and sex chromosome. Now, what is autosome and what is sex chromosome? We will look on to what is autosome and sex chromosome. Now, we can say human beings have how many? 46 chromosomes. I am writing chromosome as CH. This 46 chromosome can be written as 23 pairs. This 23 pair can then be written as 22 plus 1 pair. 22 plus 1 pair. Now, this 22 pair, which basically is responsible for the development of the body of the organism, this is called autosome. This is called autosome. Whereas this one pair, which is responsible for the sex of the organism, is known as sex chromosome. Sex chromosome. Therefore, what happens is this 22 pair of autosome will basically make the general body structure of an organism. That is, if you minus sex in a male or a female body, they are more or less identical. But now, if you include the sex, that is one pair, the sex pairs would be either XX, which will be a female, and XY, which will be a male. So this is a male and this is a female. Understood? This is a female. Now we go back. And that is I discussed about autosome. I also discussed about sex chromosome and sex determination, as whether the child is a boy or a girl. The XX factor and the XY factor basically determines the sex of the child. Then we'll try to see what are genes and alleles. Now we are trying to look onto genes and alleles. What are genes and what are alleles? Now these genes are the fragments of DNA. What are they? Fragments of DNA which basically transmit characters from one generation to the other. And now these genes, all the genes have got two different characters or two different forms of the same character. Like for example, if I say fat, the alternative form of fat will be thin. Similarly, straight hair, curly hair, blue eyes, brown eyes, fat, thin, tall, short, these are alleles. And the character which we talk about is the fatness, if I talk about the body size, that is the character, and fat and thin iris. Understood, boys? Okay, now we move on to this one is done. We will move on to dominant and recessive. Now, what is dominant? What is recessive? Now, if I give you two papers, one black and one white, which one is dominant over the other? Obviously, black is dominant over the white, because when you try to overshadow the white with the black covers the white. Therefore, black becomes dominant and white becomes recessive. Okay. Now, we will try to understand what is variation. Variation is the difference. Variation is nothing but difference. It's just the difference between the two individuals. Like you are sitting in a class, your desk partner is beside you. You and your desk partner are more or less of the same age. More or less of the same age, same sex, same section, same class, but you two do not look alike. You two look different because the genetic makeup of each of you is different from one another. Therefore, that is known as variation, and variation is very important in genetics, because if there is no variation, then the organism will look like broiler chickens, where in a coop or in a pen, if there are 500 chickens, you go to them, you pick up one chicken, look at it carefully, play with it, and then release it there. Go out of the room, come back again. You will not be able to pick that same bird again, because all the birds have the same genetic characters in them. There is no variation amongst them. They are identical. Therefore, even a small flu, like a bird flu, when it comes, it sweeps the entire population out. Otherwise, due to variation, like for example, if one of your friend is sneezing in the class, you immediately do not catch a cold, because there is a variation in the genetic makeup of you and your friend. Then we move on to Mendel's experiment. Now, Mendel's experiment is basically Mendel, the father of genetics. How did he come to know about all these inheritance? Is basically he did an experiment using different characters, different traits, and when you take one character, remember, just one character. Mono means one, di means two. So mono means one. So when you take only one character and make a cross, that cross is called monohybrid cross. We will look onto it, what it basically means. Okay. Now, if you see, we are doing monohybrid cross. So let's say I take a character black and an alternative form of that same character white. Black I represent it as capital B, white I represent it as small b. This is called the parental generation or P1 generation. When we make a cross between the two, in the F1 generation or first phylum generation, the organism will have a genetic combination of capital B and small b. Right? Then, to know what happens in the F2 generation or the second generation or the phylum 2 generation, we need to make a Punnett square. What we need to make? Punnett square. Now, the Punnett square is nothing but a box, right? Which contains somewhat of this fashion. Now, we will put these genes here, capital B, small b. That is capital B, small b here. Similarly, we put capital B, small b here. Now, when we make a cross, this becomes capital B, that is when you are crossing these two. Capital B into small b becomes capital B, small b. Here, capital B into small b, capital B, small b, and small b into small b, small b, small b. Can you see that? Now, we will try to figure out how many organisms out of the 4 different offspring born to those parents on the second generation, how many appeared black and how many appeared white. For that, we need to know, we call it phenotypic and genotypic ratio. Okay. Now, phenotypic means that what is visible. Genotypic means that is which is present in the genes. Now we will see, this is a pure black. Yeah. This appears black. This appears black, and this is pure white. Can you see this? So now, if you see genetically, how many is pure black? 1 pure black. Yeah. How many appears black? It is not pure black, but it appears black because the dominant capital B is present in both the cases, because of which it becomes black and not white. Therefore, 2 appears black and 1 pure white. Therefore, the genotypic ratio will be 1:2:1, and the phenotypic ratio will be how many black? 1, 2, 3. So 3 black is to 1 white. Therefore, the phenotypic ratio will be 3:1 and genotypic ratio will be 1:2:1, and this is the monohybrid cross. Understood? Pause. Okay. Now we will try to understand what is dihybrid cross. Now what is dihybrid cross? Now the term di means 2. So we will be taking 2 characters. Now the 2 characters would be tall and black. Can you see there are 2 characters here? One is tall, another character is black. Okay. Similarly, the LE of this would be short and white. Can you see? Short and white. How many characters? 2 characters. Now, if I represent tall with capital T, black with capital B, what will I represent? The LE of this particular character, that is short and white. Obviously, it will be small t, small b, small b. Okay, this takes place in the parental stage, that is P1. Now, when we make a cross of these 2 in the F1 generation, that is first phylum generation, what becomes is the gametes come out, and they form the individual, or something like this. This is capital T, capital B, small t, small b will be the genetic makeup of the organism in the first phylum generation. Did you understand? Can you see that? Capital T, capital B, small t, small b will be the genetic makeup of the organism. Now, how does this organism look? This organism basically looks tall and black. Why does it look tall and black? Because the gene capital T and capital B is present inside this organism, and this dominates this character. Understood? The capital T and the capital B dominates the small t and the small b, and this becomes recessive character. This becomes RECESSI. This becomes a recessive character and hides within the organism. Therefore, this organism in the F1 generation becomes tall and black. Clear? Now, we will try to understand what happens in the F2 generation. What generation? F2 generation, which is also known as the second phylum generation. And for that, obviously, we need a clear board. So, here we come. The character of the organism genetic makeup was capital T, capital B, small t, small d. Now we will try to see the different genes that come out during the gamete formation. That is, one character is capital T, small, capital T, capital B. Another one is capital T, small b, small t, capital B, and small t, small b. Can you see? These are the four different gametes that can be formed from these organisms. Now, when we put these gametes in a Punnett square, right? 1, 2, 3, 1, 2, 3, 4. Okay. 1, 2, 3, and 4. 1, 2, 4. Okay. Now we put that here. Capital T, capital B, small t, small b, small t, capital B, small t, small b. And similarly, here also. Okay. Can you see that? Is there any error? No. Now we will cross the two. That is, this cross with this crossed with this. Understood? And similarly, write it here. Capital T, capital T, capital B, capital B. Okay? Capital T, capital T, capital B, small b. Now, similarly, you can do the calculation. I'll be also doing. You can also do it in your exercise copy, so that to see if I have gone wrong, you can point me out. Capital T, small t, capital B, capital B. This one, capital T, capital B, small t, small b. Now we go on to here. This is capital T, capital B, small t, capital T, small b. Then capital T, small b, capital T, small b. Capital T, small b, small t, capital B. Then T, B, T, B. Okay, here capital T, capital B, small t, capital B. Here capital T, small b, small t, capital B. Then here capital T. Okay. Then this capital T, small t, capital B, small t, small b. Last one. Okay. Did you get me? Now, this is F2. That is phylum 2 generation of a dihybrid cross. Now, if you see it, this one is a pure tall and black. This one is 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 tall and white. Right? Capital T and small b. Therefore, it becomes tall and white. This becomes tall and black. Tall and black. Similarly, this becomes tall and white. Tall and white. Then this becomes tall, black. Tall, black. This becomes tall, black. This becomes black and short. Short. This is black and short again. Black and short. And then this becomes black and short. Then this becomes tall and black, and this becomes white and short. So you got it. Now we will try to understand the genotypic and the phenotypic ratios here again. In the genotypic ratio, the genes that are present, or we can say the phenotypic character here is how the organism appears. That means we will try to understand how many pure tall and black are there. 1, 2, 3, 4, 5, 6, 7, 8 and 9. This is black and short. So there will be the phenotypic characters would be 9:3:3:1. Understood? That is, out of the 16 offsprings born to that parent, that is 1, 2, 3, 4 and 1, 2, 3, 4. 4 multiplied by 4 comes 16. Therefore, out of the 16 organisms born, 9 will be tall and black, 3 will be black and short, 3 will be tall and white, and 1 will be pure white and short. Whereas the genotypic ratio will be too complicated to find out. Therefore, we cannot find the genotypic ratio in a dihybrid cross. Okay, so now today here what we will see is the law of inheritance. Inheritance means how your genes, your father's genes or your mother's genes are transmitted from you to you, from your parents to you. Therefore, there are basically 3 laws to be called as law of dominance, law of segregation and law of independent assortment. Now what is law of dominance means? If there are two characters, one of the character becomes dominant. So if this is a black character and this is a white, and when this is crossed, the offspring will become black. This is the law of dominance. That is, either of the characters. It can be white also. It can be white also, but either of the character, one of the character becomes dominant and another one becomes recessive. This is the law of dominance. Law of segregation. When the segregation takes place, what does law of segregation say is the gametes during gamete formation, that is, female is made up of XX, male is made up of XY. Now, during gamete formation, these X, the X and the X of the female segregate into X cells and X cell. So XX, there will be 2 eggs from the female, and from the male, obviously, the X and the Y segregate to form individual gametes. This is known as the law of segregation. They segregate. Okay. Now, what is law of independent assortment? Now, law of independent assortment means these gametes can independently assort. Like X can go and meet the X of the father, and a daughter will be produced. Or this X can also go with the Y, and a son can be produced. This is basically called independent assortment. That is, this X cell can go and mix with any of the X and the Y cells of the father, or the X of the mother can go and mix with any of the, or this is one cell, this is two. So one can go and mix with any of them, that is either A or B, or similarly two can also go and mix up with either A or B. That is, it does not have any such law to govern them so that they have a fixed link, fixed root. There is no fixed root. They can go and join with any of the chromosome to form an offspring. This is the law of independent assortment. Now, okay. Now, mutation. What is mutation? Mutation means a sudden change in the gene frequency is known as mutation. Okay. Now here we will try to understand what is mutation. Now mutation is a part of genetics, and that what it says is a sudden change in the gene frequency due to some external or internal factors. What are the factors? The factors would be, it can be radiation from alpha, beta and gamma rays, or it can be radiation of the UV radiation also, ultraviolet radiation also. The gene frequency changes, and that is called mutation. In a very simple form, I would like to tell you how it happens. Like for example, if there is a gene frequency, let's say A, B, C and D. In a very simple form. Now, the organism has a gene frequency of A, B, C and D. Now that organism, when he or she is exposed, or when that organism itself is exposed to the radiation, the frequency may alter to maybe A, C, D and B. Then what happens is the makeup of the organism changes. Therefore, certain things changes in the body of the organism, which has happened here in Kalimpong in the year somewhere around 2000. Okay, 99-2000, where a common insect to be called as glowworm. Now, what is glowworm? There are two types of worms which emit light at night. One is your firefly. Excuse me. Another one is a glowworm. And this glowworm, one is a firefly, another one is a glowworm. The firefly flies. There is no harm in that. The other one wriggles around the drains. Around the drains, you may see a small thing glowing at night, somewhere here, somewhere there. And if you examine this particular organism very carefully, the organism looks something like this. It has got a carapace, eyes, and the body is segmented into 13 segments. Okay, and it has got legs. It looks something like this. Okay, and it has something here. It may be phosphorus, which helps it to glow at night. Now, this organism, the saliva has become neurotoxic, and if the saliva of this particular insect comes in contact with human blood, the human beings are likely to die within the next 6 minutes. This insect is called neurotoxic, the scientific name. Okay, now we move on to crossing over. Now, this crossing over and linkage is not there in class 10 syllabus. It's an extra thing. Okay, it's present in the higher classes, but here I would like to share with you what is crossing over. Now, these gene frequencies, if you keep it in a vertical order, take for instance, this is gene A, B, C and D. And there is another gene. Suppose this gene has come from your mother. another gene that has come from your father. 1, 2, 3, 4 and 5. Now what happens is sometimes, not sometimes, in the baby, in the zygote, this particular gene crosses over this one and this crosses over the other one. Then what happens, after some time, after crossing over, they segregate. They segregate, and when they segregate, the genes changes. What changes? The gene changes and it becomes something like this. Like for example, this 4 comes here, this, okay. So what happens is, after the segregation, it becomes like 3, 4, 5, and C, D, E, something like this. So this is known as crossing over. Now here only one crossing over has taken place, so it is known as single crossing over. But in an organism's body, there can be multiple of crossing overs. Hundreds and millions of crossing overs can take place, because of which, even if when offsprings are born from the same parent, these two individuals don't look alike. Now, what is linkage? Let's try to understand what is linkage. Some of the genes, some of the characters are associated in between the two genes. Two genes, and when crossing over also takes place, these cannot be altered. So, from a father to a son, these characters are transmitted without being changed. Even when crossing over takes place, they are known as linkage. Understood? Like for example, father has got a tall nose. What does a father has? A father has a tall nose. A father basically has a tall nose. Then he marries some other, a lady with a shorter nose, flat nose. Then even then, what happens, as the tall nose character is linked very close to the gene, it cannot be transmitted. Therefore, what happens? That same tall nose character will appear in the offspring, and this is known as linkage. Thank you.